Students, please don't worry, you will not be on the recording. So today we have our featured faculty from our bioengineering department who are going to be talking about the research areas in bioengineering. So we only have 30 minutes today, but if you do have any questions, please feel free to drop them in the Q&A and we'll try to address them as quickly as possible. But for now, I will hand it over to Kiara and she can take it away.
All right, good morning, everybody, or whatever time of the day is, wherever you are. Thank you for joining us this morning. We're pretty excited to showcase some of the details of our program, as well as the awesome research that our faculty is doing. Just to introduce myself, my name is Chiara Bellini. I'm an associate professor in bioengineering, as well as the associate chair for research and PhD program, and joining the call, and I'll let themselves introduce themselves. Caroline, let's start off with you.
Yeah, hi everybody, welcome. Thank you for coming. My name is Caroline Pridmore. I'm the Assistant Director of Academic Programs in Bioengineering. And I'll pass over to Lei.
Hi everyone, this is Lei Wang. I'm assistant professor in the Department of Bioengineering. Also, I have a joint department in the Department of Biology. Yeah, welcome everyone. Then I will pass out to Abbas.
Thank you. Hi, everyone. My name is Abbas. I'm an assistant professor here in the Department of Bioengineering. Back to you, Kiara.
Yeah, awesome. Let's get to the next slide.
All right, so I'm going to give everybody just an overview of the department before we launch into the specifics of the research. So we at Northeastern are a top-ranked R1 research institution. We are known for our co-op program, and we are #1 in the country. And as an R1 institution, we do have an extremely high level of research activity in the department and a research enterprise that we're very proud of. We currently have 621 undergraduates, just over 100 master's students, and about 150 PhD students, with more to come in the spring semester as we do fall and spring admission. And we have 75 faculty in our department, and we have 180 co-op employees in the Boston area, with some really high-profile names and students working in Boston, but also globally. Our co-op program does stretch across the country and also across the world. A couple of alumni that I wanted to highlight as a part of this presentation is one of our master's students, Sam Johnson, who did her BS and MS with us, graduated in 2021. And she is the founder of Tatum Robotics, which was a startup which is robotics used primarily for the deaf-blind community. And this company was actually listed in the Boston Forbes 30 under 30, which we're extremely proud of her. And yes, one of our amazing alumni. A PhD student we want to highlight who graduated in 2016 is Cody, who found Imaginostics, which is a health tech startup on MRI technology, and again recognized as from a prestigious tech startup competition, and incredible things that our students have done with their degrees from bioengineering. Our location, our flagship campus is in Boston. This is where the bioengineering department is based. So on the right, this is the beautiful interdisciplinary science and engineering complex where the office is based, with a wall of labs up the right-hand side with the glass that you can see there. But our campus does spread across the country, and we have our program launched in Portland, Maine as well. We just kicked off our PhD program there, which we're very excited about. Highlights from our graduate degree, so MS and PhD. Both programs have a rigorous core curriculum based in bioengineering science, and we have a flexible selection of electives so that students can tailor their programs to the area of research that they're interested in. And we are very interdisciplinary, so combining engineering, medicine, and biology in order to solve problems. And we take advantage of Northeastern's strength in multiple areas of bioengineering, and also our location and industry and hospital partners in the area as well. And of course, on the PhD program, it's all research, but I also wanted to highlight that the MS program also has ample opportunity to get involved in research. So this can be as little or as much research as students want. So you can do a one-off directed study where you do a small project with a faculty. You can do an official master's project, which is a whole semester of work in a lab, culminating in a project report at the end. You can also do a master's thesis, which is 2 semesters working in a lab, culminating in a formal thesis defense to a committee and publishing a thesis to the university. You can do research for co-ops. You can spend your co-op time working in a lab for 4, 6, or 8 months. And we also have quite a few students who start on the MS program, decide that they actually love research, it's what they want to do, and pivot to the PhD program, which is a great way to get into research even more. So at this point, I'll pass back over to Chiara to discuss the research areas, and then she's going to talk to you a little bit about her research.
Yeah, thank you, Caroline, for this great overview. So the research is organized in 4 research areas: biomechanics and mechanobiology, biomedical devices and bioimaging, molecular cell and tissue engineering, and system synthetic and computational bioengineering. I would say that because of the inherent interdisciplinarity of bioengineering, most of us do research in different areas or cover different areas. So this is a way, this research area really a way where that we use to funnel students into appropriate coursework, but we really emphasize the interdisciplinary of the bioengineering approach. All right, so I am going to now go to the fun stuff and talk a little bit about what kind of research occurs in my lab. So I am a bioengineer through and through, meaning that all my degrees have been in bioengineering, and I have always focused on the cardiovascular system. And so in this first couple of slides, I'm going to convince you that we need to look at the cardiovascular system and why that's important. I'm sure that you are aware that cardiovascular disease remains the leading cause of death in adults 65 and older, and conversely, aging is a risk factor for the development of cardiovascular disease. And mechanical metrics have emerged as reliable indicators of cardiovascular age, which we can really use as a surrogate for biological or chronological age. In the words of Tom Sydenham, who was a British physician from the 17th century, a man is as old as his arteries are. Of all those metrics that we use to evaluate cardiovascular age is aortic stiffness, which is measured in terms of pulse wave velocity and really doubles between 20 and 80 years of age. Next, please. Of course, aortic stiffening naturally occurs with age. However, there are environmental factors and lifestyle choices that accelerate this process, causing premature aging. And so what I'm showing there is a map of the United States generated by the CDC, where each state is color-coded in a different shade of red, depending on the number of adults whose heart age exceeds their chronological age by at least 5 years. And and among these environmental factors that can cause premature aging is the inhalation of environmental pollutants. And although this has nothing to do with the cardiovascular system, so though one may argue that you have blood vessels everywhere, so everything is cardiovascular system. It's an old paper where they compare twins, of which only one of them smoked cigarettes and the other did not, and really showed how premature aging of the skin in the smoking twin compared to their non-smoking sibling. And so within this larger context, if we can go to the next slide. Thank you. Within this larger context, what my lab is doing is really trying to predict the development of, as well as optimize the treatment for adverse cardiovascular events that may occur in response to accelerated aging due to environmental stressors. And we looked at cigarette smoke, cigarette aerosol, we look at wildfire smoke exposures, and then in terms of this adverse cardiovascular event that I mentioned, we really focus on aortic aneurysms. And of course, central to this paradigm is really trying to understand how the natural aging process occurs at the different levels, from molecular to cellular, tissue, and functional. Next. So how do we do that? Part of the foundational measures that we take in my lab is trying to understand the mechanical properties of vascular tissues. Then we have built this custom built system where we can put little mouse arteries mount them on glass cannulae. We can pressurize them. We can actually extend it and measure the force. And so if you look at on the right-hand side there is a cartoon description of a vascular wall. And so that is the microstructure of it. It has 3 layers called intima, media, and adventitia. In each of these layers, there are different structural proteins as well as cells. The intima is composed of a monolayer of endothelial cells. Then we have the media, where we have elastin and smooth muscle cells. And then the adventitia, where it's primarily fibroblast and collagen fibers. And so using this device, we can evaluate the change in diameter based on an increase in pressure and build those S-shaped curves that we call pressure diameter curves. And we can also measure the change in axial stretch to reach a certain axial force depending on the luminal pressure within the vessel, which are those 4 colored curves on the bottom in the middle. And so these type of tests allow us to evaluate the passive response that is primarily associated with collagen and elastin structural protein. But next. Thank you. We can also look at how the vessel react in response to vasodilator and vasoconstrictor. And so the middle panel there shows a mouse vasculature, mouse descending thoracic aorta. Be mindful that that diameter is approximately 1 millimeter, so that we are tiny vessels that we need to tie the branches off in order to pressurize, and then we can contract. On the plot on the left, you see a decrease in diameter and then a progressive increase in response to vasodilator, and so that allows us to better understand the influence of endothelial cells and smooth muscle cells on vasoreactivity. Next, please. And so just a very quick example of an application. You probably are aware that the use of electronic cigarettes has increased in the last few years, particularly in younger adults. Because of the rapid change of these devices, we really do not have any long-term data. And so we are trying to avoid the history that happened with cigarette smoke, trying to use preclinical model to figure out what the long-term effects of e-cigarette use would be. So let's go to the next slide that I can show you some results. And so here we are using the tools that I described in the previous slide to compare the effect of cigarette smoke in orange and e-cigarette aerosols in purple on blood pressure. That's the top left plot, as well as on certain properties of the aorta of the vasculature that we are interested in, such as wall thickness, tissue stiffness, stored energy, which is a measure of the Wincastle function of the vasculature, so how well the vasculature is working, the central vasculature is working as a pressure reservoir, as well as distensibility, which is this metric that really is indicative of vascular aging. And so without going into the detail, taken together, this data really suggest that contrary to public perception, e-cigarette vaping is not safe and has dangerous repercussion on the cardiovascular system. I'll pass it to Lei.
Yeah, thank you, Chiara. This is Lei Wang. I'm assistant professor in the Department of Bioengineering. Next, please. So first, I would like to introduce the concentration of system synthetic biology in our department. And also I would like to highlight my lab's researches for mammalian synthetic biology. So I would like to see the focus of my lab is to develop mammalian synthetic biology tools for biomedical application. I think everyone here, one back please, sorry. Yeah, everyone here, we have used or heard about the synthetic biology products, for example, like the COVID vaccine and the CAR T-cell therapies. And then, so that means, yeah, we already enter in an age that synthetic biology really plays a really critical in our already daily life. In addition, I would like to see the location of Northeastern University, Boston. That's the global hub of synthetic biology research, and also hub of the synthetic biology-based startups. So now I think here is really great space to study synthetic biology. Then I will go deep into what my lab is doing. So we are using a bottom-up strategy to help develop tools for biomedical application. First, we are developing molecular synthetic biology tools. In that way, we develop new technology and tools that is generally applicable to a lot of applications. In addition to that, we will go a level up that will be go from molecule to cell. Then we can use these tools to really engineer cells. For example, that has application in tissue engineering and regenerative medicines. In addition to that, after we engineer cell in a function, the way we really want it to be, then we can go to another level like organ or even whole organisms. Based on that, we can get new knowledge how to advance for advanced human health. Next, please. So the hardcore of all the strategy we have, the bottom is the synthetic biology. What's synthetic biology and how it works? For synthetic biology, it's a kind of relative new field. It's about 10 years age. So we already observe that in naturally biological system, there are already a lot of elements works in some way, but in different species, different contexts. As synthetic biologists, we just bring different tools in different organisms and repurpose them into a way we really want to have and to advance human health condition. And how can we do that? We follow a design, build, test, and learn cycle. First, we design some circuits. We can use like some tools like artificial intelligence to like a huge library based on the knowledge we have already for different biological parts in different species. And then we will build the molecule in a way we can encode into a DNA or RNA. So now we have the multi-clone system that really can speed up the built-in process. Before that caught a very long time. Now it's very efficient. Next, we can test the encoded DNA or RNA in the level of biological system we are interested. For example, like E. coli or cell lines or even the whole organism. And after these 3 steps, we really can get a result and learn what can we optimize for next cycle of experiment. And then after several iterations, we will get optimal design for a specific goal to tackle the biological question or the biomedical issue we have currently. Next, please. So the question I'm interested about is the cell state transitions. I think everyone here, we all begin with a single cell, but later we go through a lot of developmental state transitions. We become a human being. We have over thousands of, even 200 of cell type. Next, please. Yeah, we have over 200 cell types, and then we become organism, become over trillions of cells. And it's very interesting that all these cells, they are not similar. They have their own function and their own cell types. Even though we know a lot already due to the progress of science, but still we do not fully understand how a single cell can differentiate into these 200 different human cell types. But if we can really understand this very well, that can significantly advance our understanding and our technology for regenerative medicine and human disease model, even cellular therapy and human in vitro models. Next, please. So if we have a close look about different cell types inside human, if we see that in the DNA level, they are similar in terms of DNA, but the most different part happens in the post-transcriptional level. Specifically, that means RNA and protein. That's the difference of RNA and protein really shape a cell type like a neuron and muscle cells and immune cells. Among all these post-transcriptional regulators, microRNAs, that's this year's Nobel Prize winner, that's microRNA, plays a central role in cell lineage determination by fine-tune gene expression. Next, please. So in our lab, we already developed a microRNA sensor based on the endogenous microRNA species that can tell the cell specificity, that tell the cell type. And this technology has already been patented and currently has been applied in a startup in Boston. And further, we can use this sensor to engineering cell type. As we see for cell differentiation, there are different step-wise the differentiation, and they can differentiate into different cell types by programming cell and sensing their current cell state and express specifically the transcription factor can guide cell differentiation. We are able to program cell in an automatic way. For example, we are able to differentiate iPSC cells into the hematopoietic progenitor cells. In addition, we have the laboratory, we have the platform really can build like a cancer on chip model or organ on chip model. Together with this, all the platform we already have. Currently, we are able to have the libraries to really screen all the human transcription factors and screen the function of all human transcription factors. And then we can get ideally any cell type we want for the purpose. For example, iPSC-based cellular therapy, organ and cancer on chip models, and even for the in-field cell engineering. Next, please. Okay, and then, I hope I introduced about what my lab is working on. And if you want to know more about the research of my lab, please scan this QR code. And I would like to hand over to Abbas.
Thank you, Blay. Next slide, please. That's a very jarring image. So my lab, we build and apply optical imaging technologies with the goal of trying to understand brain function at the cellular and microvascular level. So this image that you see here on the right, it was taken with 2-photon microscopy, and it's in the live mouse brain, and we're looking at of green labeled astrocytes, and the red label is blood plasma. So it illustrates the microvasculature, and we can actually visualize the passage of individual red blood cells. Next slide, please. So the motivation behind applying optical imaging specifically, next. Diseases like Alzheimer's disease, Parkinson's disease, stroke, they all their onset and progression involve like quite a multifactorial processes affecting a number of different mechanisms in the brain. So there's deterioration of neuronal signaling. There's also concomitant change in energy metabolism that has influences on blood flow. There's also inflammatory processes that develop over time. And our goal is to understand very precisely how these all manifest and then progress throughout the variable trajectories of stroke, Alzheimer's disease, and any number of different other brain pathologies. So optical imaging, its key strength, in addition to being non-disruptive. It allows us versatility and the ability to look at multiple seemingly orthogonal signatures of brain activity. Next, please. The other underlying motivation is to actually get a better understanding of the signatures that you're measuring with more translatable opticals. So this image of the brain here, that was collected, that's in a human brain, and that was collected with functional MRI. I do not do this type of imaging. It's arguably one of the most transformative technologies that have come out to better understand the actual, to visualize activity in the human brain. However, those red and blue pixels that you see there, they're indicative of changes in blood oxygenation, blood flow, and blood volume. And so those can be considered somewhat downstream consequences of changes in neural activity and the associated partner cells that are communicating with one another to actually give rise to dynamic changes in the microvascular activity that fMRI can see. So in essence, the images that I collect look more like the images that are on the right, and the goal is to understand in detail the processes going on between these different very vital cells and structures in the brain, and how they actually contribute to the overall messaging, the overall image that you see on the left. I'm going to try and speed these things up in the interest of time. So next slide, please. One project, I'm very interested in looking at the benefits of non-pharmacological treatments in an effort to mitigate the onset of some of these very dire threats to the human population. So Alzheimer's disease, I'm very interested in looking in ways to try and just curtail the threat that it poses to the population. So we have this long ongoing project trying to understand the benefits of physical exercise and how that can help reduce the likelihood of dementia in later life. Again, that's that fMRI image, and the basically each one of those red and blue pixels is indicating at each one of those pixels you see it. You actually measure a time resolve signature similar to the graph that you see in the bottom plot. Can you go to the next, please? With our multifocal microscopy and oxygen tools, we were able to measure those signatures in individual microvessels at different layers in the cortex, and we can measure those longitudinally. And our study is indicating that physical exercise does have some pronounced benefit for preserving microvascular oxygenation and metabolic function. It's promising, but it's not a panacea, say. So that work is still ongoing. Next slide, please. Can you play the movie? So here we're looking at when you see the yellow timestamp, that corresponds to a time where we actually induce a functional stimulation on the mouse, and so you see this increase in brightness in the green, and then you see a change in the red arterial diameter that corresponds to like the arterial dilation. So next slide, next please. Those green cells actually play vital roles in inflammation and in blood flow. So we've got a study that are trying, or a big thrust of the lab is trying to understand the connections between neuroinflammation and blood flow, and its consequent influence on the readouts that you get from translational imaging methods like fMRI. Next slide, please. So those are some of the biological questions that we're asking, but in terms of engineering, we're developing new ways to access deeper structures of the brain. This is a technique that we're adapting. Next slide. Basically, you can implant a lens into the deeper structures of the brain. This was a published paper that came out a few years ago where they developed a very what's called a cannula, so that you could temporarily insert a lens in there into the brain and an image, and then remove it when you don't need it. Next slide, please. We've adapted the cannula technique so that we can not only can we visualize things, but we can also deliver pharmacological agents and contrast agents into the hippocampus. So this is opening up new opportunities to measure a number of different indicators of brain function in deeper structures like the hippocampus, which are some of the earlier, more vulnerable targets of Alzheimer's disease. Next slide, please. Another big thrust currently going on is we're actually incorporating other optical technologies besides two-photon microscopy. So we're building up the system where we can, rather than measuring things with microscopic detail, we can measure things with mesoscopic detail. Next slide, please. And the ultimate goal will be to actually measure things on a larger field of view with slightly compromised spatial resolution. But we'll be able to see signatures of hemodynamic activity and cellular activity. And more importantly, we'll be able to very rigorously assess connections across different regions of the cortex. Several diseases and other disruptors of brain activity having a pretty pronounced influence on these things. So there's a whole host of studies that I'm very excited to embark upon once we've got this system up and running. I'll leave it there, I guess, and pass things back to Caroline or Kiara. I'm not sure, but next.
Thank you, everybody. And thank you for all your questions throughout the presentation as well. If you do want to learn more about our graduate programs, you are more than welcome to reach out to myself, to Kiara. If you want to learn more about the research, reach out to Bas and Lei and Kiara. And one question which we haven't been able to answer yet is about if we have any open data sets or a code repository for performing own analysis, or if not, if we could suggest some ways that someone could do that.
I can answer that. So most of our research is funded through federal grants, and federal regulation indicate that you gotta share your data. And so there are some repositories that are managed by mostly NIH, where you can find raw data associated with some of these projects. So I would definitely check on their website.
Can I also answer that? A lot of the work that you've seen here hasn't been published yet, so it may not be available until, well, it could be a while, but eventually it will be made available.
Great. All right. Well, thank you everybody for coming.
Thank you too to our faculty. This is a very busy time of year for them, so I really appreciate them joining today, and I appreciate all of the students, prospective students joining from around the world. If you have any questions, I did drop the COE admissions email in the chat. Please do feel free to reach out to us, and we're happy to connect you with the person that may be able to help you. But thank you everybody for joining today, and have a wonderful rest of the day.
Thank
Thanks,
you.
everyone.
Bye.